using stem cells for human therapy, cell replacement, drug screening, and disease modeling. I'll give you a little brief bio. I'm just looking through you there. Dr. Sullivan was born in Cork City. He graduated from University College Cork with a degree in biochemistry. Following this, he went on to study for a master's in research from Trinity College Dublin. He then went on to the Roslin Institute in Edinburgh, where he earned his doctorate under Professors Jim McQueer and Ian Wilmot, which are two of the scientists responsible for cloning dive sheep. There, Dr. Sullivan became one of the first researchers in Europe to culture human pluripotent stem cells, and you'll find out more about this for Dr. Sullivan. He moved to the University of Cambridge, where he got an Isaac Newton Trust Fellowship. He subsequently went on to work as a research fellow at the Harvard University, where he earned a stem cell research fund scholarship. So there you go, that's his bio, and I'll hand you over to him now. So.
First of all, just let me say thanks very much for coming in when the weather is so good outside after the last 2 Irish summers. I think you're making a specific, a very, very big sacrifice to come in when the weather's this good. So thanks very much. I've been back in Dublin for the last 6 months now, and it's really very encouraging. I've been away for a long period of time, and coming back to Ireland now, it's really striking how much more people are becoming engaged in scientific topics in Ireland now. You've got, like, for example, this Alchemist Cafe. You've got things like Science Spin magazine, and also you've got a lot more science on the TV. And what I really like is the Science Gallery, where Cormac gave us a great talk on the Big Bang last week, or 2 weeks ago. But the Science Gallery specifically designed to bring science to a broader audience, because I think in this day and age, scientists, it's not always clear to people why scientists want to do certain things that they want to do. And this is particularly in my field of stem cell research, where there is, every day there is like a different story or a different spin and a different misinformation kind of in the newspapers or in the media. And I wanted to touch on that towards the end of the talk, that really, media is changing from print to digital, and that big upset now is changing dramatically how we get our information. And when we're looking for professionals, it's not always easy to find the professionals versus some person who just likes to rant in an unsubstantiated fashion on the internet. And this is, I think, pertinent very much now, because we have a rising degree of stem cell fraud. You have people making money off people who are in desperate situations, who have either sickness themselves, or they have a relative who has a sickness, and they log on to a website, and the website tells them that there's a cure available, when I think the medical and scientific consensus is there is no such cure as yet. So excitement that really science is getting deeper into the public consciousness, and that's great to see in Ireland. It really is. So well done on the Alchemist Cafe for taking part in that. My curiosity as a scientist is more or less centered on the regenerative medicine and developmental biology field. So I'm kind of interested in how a fertilized egg becomes a full sentient thinking human being, how cells know what to do at the right time. How does a neuron know to become a neuron? How does a liver cell know to become a liver cell? And the great orchestration of development is intriguing to me, that you can get such complexity arising from initially just one fertilized egg. So, also of interest is the idea of regeneration, how we regenerate ourselves. For example, like in our gut, for example, the gut regenerates itself almost on a daily basis. The lining that our food passes through and gets absorbed. And that's very, very interesting that, you know, when you take, say, for example, lower vertebrates like a salamander, if you cut a limb off a salamander, the whole limb regenerates. And, you know, there's this great interest in if the lower vertebrates have this capacity, why doesn't it still exist in higher vertebrates? And whether actually that capacity is still there, but something is inhibiting it. Like, for example, something like scar tissue formation interferes with some regenerative capacity that's already underlying. So these are really kind of big questions relating to either directly or obliquely to stem cells. Stem cells are a unique kind of cell that have two characteristics that make them extremely interesting to biology, both as a tool, but also as a cellular identity. They have the capacity not only to self-renew, so like one stem cell can become two stem cells, can become four stem cells, and so on. And this, I suppose, a capacity to start off with a small amount of material and augment that material is very, very important for a lot of kind of experiments that basic science wants to do, but also is extremely important for having enough material to effectively treat a patient with a disease. So, self-renewal. The second capacity of the stem cells that make them remarkable is that they can also form cells fated to different functions. So, for example, if you take a blood stem cell, known in the trade as a hemopoietic stem cell, it can divide to give you more hemopoietic stem cells. So you start off with a small number of cells and you can augment that number. But also, they can make all the cells of the blood system. So, you know, this is extremely. These two characteristics make them extremely useful for, say, conventional stem cell therapies involving leukemia or rare blood diseases. Like, for example, way back in the atomic bombs landed on Hiroshima, there was a lot of interest in that. You know, apart from the initial blast that killed lots of people, subsequently people died a few weeks later. And what they basically came to understand is the radiation was killing fast proliferating cells that were essential for the survival of the people. So they did more research into this and did several very interesting experiments with mice, where they took bone marrow from a normal mouse and then transplanted that into an irradiated mouse, and they found that the irradiated mouse was rescued. It didn't, after being exposed to the irradiation, die anymore. And we now know that is because there are several stem cells present in the bone marrow that replaced the cells that were initially damaged and died after the mouse had been exposed to radiation. So these stem cells are hemopoietic stem cells or blood stem cells, and also mesenchymal stem cells. So between the two of them, they repopulate a system like the immune system and the blood system, which are essential for continued survival of the mouse. So, in many ways, stem cell therapy is not new. It's now 50 years we've been transferring bone marrow cells into patients in one form or another, and even now, after 50 years, the number of diseases that we can cure and treat is still very small. And the reason for this is that there are a lot of questions that are still unanswered in stem cell biology. One of the things is that stem cells, such as adult stem cells like blood stem cells and mesenchymal stem cells, they're very hard to find. They're very hard to isolate from other cells that look sort of similar in the bone marrow but don't exhibit the same properties.